Hello, my name is Sydney Abbott, and this is my oral report for ECCB 205 with Dr. Grace. I went to the plenary talk, The Genetic Structure of Townsend Long-Eared Bats, presented by the undergraduate student Caitlin Sanchez. The long-eared bat, also known as the whispering bat and the lump-nosed bat, was the main subject of this presentation. Now for some background information on these bats. Located in the western part of the U.S., Townsend long-eared bats are often found living in caves or abandoned buildings, and return to these same places year after year. Additionally, these bats are very matriarchal, leaving the females to raise the young in the summers while the males disperse. In the fall, these bats move into hibernacula, which is a shelter occupied during the winter months by a dormant animal, such as the abandoned caves and buildings I mentioned before, where they will stay until the springtime. Unfortunately, these bats are a species of special concern. According to the California Department of Fisheries and Wildlife, this species has started to exhibit a steady decline over the last few decades due to three main threats: white nose syndrome, habitat disturbance, and pup abandonment. In this presentation, Caitlin explains that her project is collected data of her population and genetic differentiation rather than an experiment. Her objective is to determine the genetic structure of the hibernacula colonies, as these colonies are the main reason white nose syndrome spreads so rapidly across this species. She surveys multiple breeding colonies from the Sierra National Forest together, as well as an individual colony from the Sierra National Forest as well. To fulfill her objective, she uses 4 different methods: microsatellite markers, Genus, a data calculation software, Elequin, a software for population genetics, data analysis, and Structure, a software package for using multi-locus genotype data to investigate population structure. Using these methods, she calculated the FSI significance, which was less than 0.01 of amitochondrial DNA. The measure of this FSI significance is the measure of genetic differentiation or distinction. From these results, she was able to conclude that the colony that was studied was genetically healthy, and the bats within the colony do not appear to be genetically isolated from each other. Later in this presentation, Caitlin explained that examining sex-based dispersal more in depth and expanding the survey area were going to be the next steps in her research. Although this planetary talk was very informative, I will admit there are some things I did not understand, but this is mostly because I currently don't know a lot about genetics. Because I am so unfamiliar with advanced genetics, I didn't feel qualified to ask any questions. However, besides some slight confusion, I found the talk to be very interesting. It was really refreshing to see someone speak so passionately about a topic they care about. And although these topics covered in Caitlin's research were much more in-depth, I was excited to see how the basics of genetics that we've learned in our class can be applied to important research such as this. Thanks and gig 'em.